I think it's right time to start. I think everyone is into the meeting. So hello everyone. Wish you a good day to all of you, and welcome back to Palaeopax weekly seminar. And today we have a wonderful speaker, Gustavo A. Ballen from Sao Paulo State University, Brazil, and he will be talking about paleotheology and Neogene Palaeoceanography of Northern South America. So a little bit about today's format of seminar. So we'll follow the welcome and announcement, which is happening right now. And then Gustavo will be talking for 30 minutes, followed by moderated question and answer. And please don't forget to send your question to the Paleopax host, who is today Gwyn. And Gustavo also agreed to stay with us for tea time. So where you can discuss a little bit more in relaxed setting. So after the talk, at the end of the seminar, please stay with us for the tea time. And a little bit about the Paleopax housekeepings. So Paleopax values the participation of all folks interested in paleosciences. Please remember to abide by our code of conduct during the seminar. And you shouldn't be unmuted during the talk. If you do, please mute yourself. And please don't forget to ask the question by chatting to the Paleopax host or by using the raise hand function. If you face any technical issue during the talk, please reach out to our Paleopax host, and we also have closed captions built into the Zoom. Please use the CC button to show or hide them, and we promote the work of early career researchers. So if you know any outstanding ECR, so please nominate them, or you also can nominate yourself, and the link will be dropped out into the chat box. And we also get weekly feedback from the participants for the demographic information, which is completely optional and anonymous. So, and link will be provided into the chat box for the feedbacks. And I would love to introduce our speaker. Gustavo has done his bachelor from National University of Colombia, and he also has done his PhD from Museum of Zoology, University of Sao Paulo, Brazil, followed by a postdoc from Queen Mary University of London. And currently he is doing another postdoc from Sao Paulo State University, Brazil. So with this, I invite Gustavo for his talk. Gustavo, now screen is yours. Please, can you start sharing your screen?
Thank you very much, Salman. And thank you everyone for sharing this time with me. Right. So can you see my screen?
Yeah. It's great. It looks great.
Right. Okay.
Let's go ahead.
So thank you, everyone. So today's talk is a combination of results from a couple of projects I've been doing during the last years, which includes also what I did during my PhD at the University of São Paulo. So one of the big questions in biological sciences in general and environment is biodiversity. And we have in this picture here a map with the so-called biodiversity hotspots, which are regions of the world with an outstanding biodiversity. And we see here in South America, we have some regions which are considered hotspots. Especially this region, this vertical region here, which is, we will see in a minute what it is particularly interesting and special for us today. This is South America for those of you who are not familiar with the geography of the continent. So in South America, we have this very, very long mountain chain, which is called the Andes or the Andean Cordillera or the Andean Mountains. And it essentially separates between two main regions, east and west of these mountains, right? So this huge region, which is east of the Andes, and then this tiny region, which is west of the Andes. We can close up a bit on what we consider here northern South America, which is essentially this region north of the Amazon main channel. So we are concerned with the paleogeography of this region here, which involves the Andes, the Panama Isthmus, the regions west and east of the Andes, the Caribbean Sea, Pacific Ocean, and right, so this region. Okay, so but why are the Andes so interesting? In this work by Antonelli and colleagues in 2018, they identified several interesting questions for what they call transdisciplinary biogeography. Three of these are especially interesting here. One being the isthmus formation and biotic interchange in this area. Another one related to river dynamics and diversification, which is mainly in the Amazon. And finally, mountain uplift and adaptive radiations. This region here, and especially these three processes, are especially because they are linked in geological ways. So the paleogeography and the tectonic history of Northern South America affected all these three. So on one hand, we have the cluster of the isthmus sometime during the Neogene. On the other hand, we have the sequential uplift of the Andes from south to north and how it affected the drainage reconfiguration in Northern South America, of course, also associated with the formation of big lake environments such as the Pebas system and the influence of marine transgressions. So essentially, we have this mountain chain here, but it is interesting because it separates drainages once it's completely formed between things that drain to the east and some others that drain to the west or either into the Pacific Ocean or the Caribbean Sea, right? And this happens because the Andes uplift. Now, why fishes? So fishes are not one of these very charismatic groups, such as birds and mammals, but they are especially interesting in this part of the world because of their enormous biodiversity. And this is not just in number of species. For instance, one of the most diverse groups, which is siluriforms or catfishes, has worldwide more than 3,000 species, and they are particularly specious in the neotropics, but also because in general, not just catfishes, but all of the freshwater fishes in South America are highly diverse in ecomorphological ways and also in species richness. And here we have a small sample of the enormous variety of shapes and forms of living. And of course, we cannot see their for instance, diet variations, but they are very, very, very diverse. So they essentially live in all of this continent in the fresh waters, and they also are very spacious. They have huge amounts of species, especially in regions in this upper map here with warmer colors such as red. That means higher numbers of species, and this happens in the Amazon, the Atlantic forest, the Guyanas, and this region, which is also Northern South America, also affected by the Andean uplift. But not just that. It also happens that if we plot the percent of species which are unique to the drainages we're interested in, we see high proportions of endemism, especially associated to the Andes. So something really interesting happens here. It's not just that we have lots of species, but also a lots of species that are only in those drainages. Why this happens? We can see more or less the same information again in these two maps. Here I'm highlighting the Andes. The region east and west of the Andes, and a couple of interesting plots. In this one, we are seeing the famous species area plot, in which larger areas have more species, which is what we expect. So small drainages, which are west of the Andes, in comparison with the Orinoco, for instance, which is a huge drainage here, it has more species. But when we plot the number of endemisms, which is here, against the richness, we see these small drainages have lots of species which are unique to those drainages. What this means in biogeographic terms is something like this. We have lots of species here, but we have some species with, or some groups which are shared between these two areas, east and west of the Andes. And this is especially important because those species or those groups which are west of the Andes have relatives east of the Andes. So potentially they can help us understand how these areas were separated through time. So this is our study area, which is northern Colombia, and this part here is Venezuela. So here we studied 3 localities, which are 2 of them in the Guajira Peninsula in northernmost Colombia, and another one in the city of Corozal in northern Colombia too. And these are the Castilletes and Guare formations in the Guajira and the Cincelejo formation in Corozal, which are essentially Middle Miocene in the case of the Castilletes formation, and Pliocene, both the Cincelejo and Guare formation. So once upon a time, these regions were more similar to what we see today, for instance, in the Orinoco drainage or the Amazon, that is forested areas with large biodiversity, lots of water, animals. But what we see now is this, which is essentially a desert, which is really good for finding fossils, right? But it tells us about the dramatic change in environmental conditions that happens between the Miocene to Pliocene, and today. There's been a lot of contributions in different vertebrate groups, and fishes were one of the least studied groups, which are what we are concerned here. So essentially, we have this situation that where we separate drainages as the Andes are uplift, and potentially we can trace this separation through time by studying the fossil fauna at those times, right? So this is what we are interested here in this work, whether these 2 regions were still connected by the Pliocene or whether they were separated by the Middle Miocene, which is the historical way of thinking that postulates that the Andes separated during the Miocene, and therefore during the Pliocene they were already separated in eastern drainages, east and west of the Andes. So we are tackling this pair of hypotheses. What do we need to test them? First, we need fossils from around those ages, but we also need comparative anatomy and extant representatives for identifying these fossils. Also, we can use divergence time estimation for cis and transandean, or these are east and west of the Andes, sister pairs, and their estimated times, so that we can try to use statistical inference for inferring this the origin of this phenomenon in time. And we need those statistical methods for estimating origination time of events, and finally a computational implementation of said methods. Then we started collecting fossils. This is in the Guajira Peninsula and this in the Cincelejo Formation. And we collected either by hand-picking surface collection, but also by dry screening, so that we could concentrate micro and mesh fossils and study them. How do I identify those fossils? So first thing, we had to develop a lot of comparative knowledge, which ended up being published, for instance, in this work, which is a standardized terminology of spines in silviforms. And this happens because spines are really, really common in the fossil record, and they are really, really overlooked under the premise that they are not informative. But this is not the case. Quite often they have very, very structured variation that can help us identify different taxa. So when we combine our knowledge on comparative anatomy and comparisons with the modern fauna and our fossil specimens, we can try to identify them. And this is what we find. In these three contributions, we have been publishing several taxa, mostly as fragmentary findings, which is essentially what you find in these regions. This is not like a exceptional preservation, but still is very informative about what kind of fishes were there during the Pliocene and Miocene, right? So what's the deal with this with these fishes we're finding? Are these occurrences unexpected? They are sort of unexpected because if we plot the distributions of these groups in recent times, we find clearly those groups being restricted to the Amazon, Orinoco, and Guyanas, which are east of the Andes, and no occurrences at all west of the Andes. But these are our fossil localities, which are west of the Andes. And by those times, Miocene and Pliocene, we are finding fossil record of those groups across the Andes, which suggests us that there should be some form of drainage connection by that time, and therefore not the whole Andes were uplift. This also happens in this particular species, which we know is this species because of comparative work, but also phylogenetics. And this species is restricted to the middle and upper Amazon basin nowadays, but we have fossil record here in northernmost Colombia. When we have the possibility of placing those fossils in a phylogenetic context, they usually tell us interesting stories. For instance, in the case of Fractocephalus from the Cincelejo Formation, it shows a clade with the fossil specimens and the extant species to the base of all of them. So this is essentially something related to the other fossil Fractocephalus. And in the case of Hydrolicus, this one, it helps us constrain the identity of the fossil to a extant species, reinforcing the conclusions before. And of course, we here use Bayesian methods for estimating or measuring uncertainty in our phylogenetic estimates. Now, what happens when we consider the whole fauna is something like this. When we use hierarchical clustering with fossil assemblages during the Neogene here across all of South America, we don't see this correspondence between areas east and west of the Andes that we do when we do exactly the same analysis with modern assemblages, essentially with the same information, species occurrences across the different drainages in all of South America. And here we have this cluster, which is the Transandean cluster, and then this huge Cis-Andean cluster. The greenish lines point toward the Transandean one, and we see it's sort of scattered across the hierarchical clustering of fossil assemblages. That is, by the time we had these assemblages, probably there was not so strong geographic structuring or clustering, which points again to the possibility that we had still drainage connection, or also we had other processes determining the composition and similarity or relationships between these faunas in the fossil record. What we interpret with these results is something like this. We here have the Andes, but this region here, which is called the Garzon Massif, which is a tectonic block, is not completely uplift, therefore allowing this branch, which originates west of the Andes, to come across and drain this region, which is now called the Magdalena Basin or the Magdalena River Basin. And this explains the drainage connection between areas east and west of the Andes. Some people don't like the idea of having the Merida Andes and coastal Cordillera to being not completely uplifted by that time, and that's absolutely fine. It can be that those regions of the Andes were not completely uplifted, but this might still explain how these things were still connected. Now we're contrasting the information from the fossil record with divergence time estimates. And here we have some simplified scenarios. So we have an area, for instance, South America, and then by a tectonic process, we are building mountains here and separating in what was an ancestral area into two daughter areas. And then once this happens, the biodiversity reacts by speciating, by vicariance mostly. When this happens and we survey lots of groups from birds and plants and mammals, but also fish, by collecting those informations that is at what time we have nodes with east and west of the Andes, pairs east and west of the Andes, and then do statistical inference on those data, what do we find? And for this, we developed two methods. One of them is the x-intercept inference, which is essentially instead of being interested in estimating the y-intercept and slope in a regression model, here we have the pattern of cumulative distribution of occurrences in time. And then we can estimate the x-intercept, which is another parameter here, and build confidence interval on them. Right? So if this is our pattern of occurrence accumulation of cis transcendent pairs, we can estimate when this started to happen. On the other hand, we can think of these occurrences in time, as if these were stratigraphic intervals, that is, collections of time occurrences, but for which the origination and extinction times are not known, are not observed, so we can use statistics for estimating them. And these methods were implemented in this package, which we call Tibia, which does some other stuff for Bayesian evolutionary analysis. So I invite you to take a look to the different possibilities that this package gives for us. But what's interesting here is we use this data set as an example in this package for illustrating the estimations using these two methods, and they find some support to the idea that the origination of these cis and transcendent separation events happens somewhere around the Miocene to Pliocene to the Pliocene times, right? What is inconsistent with the idea that the Andes were uplifted by the Miocene. Now, taking a look again at stratigraphic intervals, there's this method which was recently implemented in this Julia package. And here we are interested in using again the idea of stratigraphic intervals, but not just for time occurrences, but instead to model the fossil record of complete clades so that we can use the estimation of the origination time, the distribution associated to that estimation, as a prior for node calibration in divergence time estimation. So we did this for 5 clades of Carengarians, which is this marine group. And we did this because we were interested in seeing what was the relationship between the evolution of this group and the closure of the Panama Isthmus. So we collected all of the fossil record for these groups, especially barracudas and centropomids, which were our groups of interest, but also for other families or superfamiliar groups, and used stratigraphic intervals for estimating the origination time of those clades. And this way, we escaped the arbitrariness of specifying node calibrations by just picking the oldest fossil and then setting the maximum of that interval arbitrarily, probably using another previous work, right? Because here we have a proper distribution that we can just input as calibration in divergence time estimation. What we find here, and also using biogeographic modeling, is that as is suggested in some papers on the Panama Isthmus closure, there seems to be some support to the idea that this happens during the Miocene and not during the 2 to 3 last million years during the Pliocene, as is usually told, right? And this is because we find some nodes for which we have high evidence of ancestral either Western Pacific, Eastern Pacific or Western Atlantic or Caribbean ancestral area. And this happens, it's important to note, on the apical part of the tree, which is not so affected by the uncertainty associated with the basal part of the tree. That is, the farthest we are from the tips, the more uncertainty we have in the ancestral state reconstructions. This also happens in biogeography. And here we have some evidence that this might be happening during the Miocene. So to recap, some ideas I want to reinforce. One is that fossil fishes are a rich source of information on paleogeography. Another one is the integration and comparison with the recent diversity are essential for this study or for their study. That fossil record and molecular evidence do not necessarily point towards different explanations because those phylogenetic analyses that we were showing were essentially combining DNA and morphological information. And it is possible and desirable to combine fossils and extend datasets with molecular and morphological datasets in total evidence approach. Persistent drainage connections across the incomplete Andes by the Pliocene were in place instead of the Middle Miocene. That is, our estimates of a younger persistent connection than it was previously thought, but also complete closure of the Panama Isthmus sometime during the Miocene that is older than is usually thought. I want to thank especially FAPESP, which is the funding agency that have supported most of my career since my PhD scholarship to my current postdoctoral fellowship, but also the institutions that have helped me through the years in logistic terms of other ways, such as the São Paulo State University, the Museum of Zoology of the University of São Paulo, Smithsonian Institution, Queen Mary University of London, the Instituto de Ciencias Naturales at the National University of Colombia, and the Universidad Javeriana in Bogotá. And thank you very much for your attention, and I'm happy to take any questions.
Great. Thank you. Thank you so much, Gustavo, for such a wonderful talk. Thank you so much. I think now it's time to cook for question answers. I request all the participants, please send your question via chat box or please by using your raise hand function. Please send your question to the Palaeopax host. So with that, I think we already have few questions. Okay, so first question is here for you, Gustavo. Great talk. Do you find how do the finding from the fossil fishes compare with what mammals or other vertebrate groups suggest about Neogene Palaeoceanography in northern South America?
Thank you very much for your comment. Well, that's actually quite interesting because regarding mammals, we have really strong how can I say that? Faunal turnover, right? So, for instance, when we look at the mammals of the Miocene, they are drastically different, like completely different groups, extinct groups that are no longer around. And I'm not aware of any species or genus which has fossil record reaching the Miocene in mammals, but that's definitely the case in fishes. So we find some genera which are present up to the Oligocene. Right. So in that sense, we have less turnover, and therefore I'm not aware of mammals being used as evidence, for instance, of past connections. Of course, other than when what we see, especially during the Great Biotic Interchange, right? So in that sense, I see other groups being used also for questions in paleogeography in that sense. Now, if the question is related to the timing of drainage separation, I would say in terms of fossils, probably not, but definitely those estimates from divergence time estimation, which are also from mammals and other groups, such as plants, point towards a younger separation of drainages. So I think that's how it compares. But there's an interesting case of I think it's turtles. Yeah, so we have fossil record of the genus Chilus, which is the Mata-Mata turtle, during the Miocene in Transandean localities. And this will be essentially the same pattern, right? I can't remember quite well what the turtle composition is in the Ware formation, which is a Pliocene.
Oh,
I'm sorry, but
okay.
I think there are some components which are shared with faunas which are currently restricted to east of the Andes.
Okay. Thank you, Gustavo, for a very fair answer. I think we have another question here. Thank you for such a wonderful talk. Among the fossil formation you studied, which one provided the strongest evidence for freshwater drainage isolation during the Neogene, and why?
Well, actually, I think it's quite the opposite. I think all of these formations, which are Miocene and Pliocene in age, point essentially to the lack of isolation. That is, that drainage connection was still persistent up to the Pliocene, which implies that not all of the Andes were already uplifted, separating drainages east and west of the Andes. And this is interesting because this is not a new idea in the sense that the work of John Lundberg during the 90s pointed to the essentially the same evidence using the Middle Miocene La Venta fauna as evidence of connection across the Andes, but during the Miocene, and then he didn't have any access to Pliocene faunas. But this has been sort of accepted for a long time that there was a still connection during the Miocene. And then we are essentially finding the same patterns, but instead during the Pliocene in younger faunas. So it's essentially just like pushing to the present this last phase of connection.
Thank you. Thank you. Thank you so much, Gustavo. I think
Thank you for your questions.
we have another question. Yes, sir. How have you gained the expertise to interpret the paleoceanography and tectonic as a paleontologist, and were there any challenges?
Absolutely. There are lots of challenges. Well, I didn't mention about the details, but my my background is in biology, so I'm not actually a geologist. But I started studying fossils during my undergrad, and this was because a fantastic opportunity of working with Carlos Jaramillo's group, sorry, in the Smithsonian in Panama. And Carlos is a geologist, and he has a huge group of a huge multidisciplinary group. So I had access to geologists and other biologists, and this helped me gain first-hand experience in field geology, but also in the literature as related to Andean uplift. So this helped me a lot. That's not to say that I'm an expert in the geology and tectonics of this part of the world, but definitely that helped a lot. And I see this is something that tends to be lacking in when we see the literature written by biologists that didn't have this opportunity of interacting with geologists. So that's the reason. I just had access to marvelous geologists.
You have good experience. and you have covered very like broad aspect of like tectonics, paleontology, everything you have covered. Thank you for your answer. I think before going to one last question, I just request audience, if you have any question, please drop it into the chat box. So here we have, I think, last question. How can Bayesian statistics be applied to other fields, say musicology? What are the strengths and limitations?
Well, thank you very much for the question. I accept it's sort of quite far from the topic of the talk, but the reason of this question is because I have also published some works which are in the intersection of phylogenetics and musicology, essentially using Bayesian methods. And the reason I really like, and this is obvious from the presentation, I really like Bayesian methods, is because not just because help us, they allow us to incorporate previous knowledge, which is like the standard reason people say they like Bayesian inference. But the one I really like is that it's a very coherent way of representing uncertainty. So when you use Bayesian methods, we don't have just like a point estimate like we have in frequentist approaches, but rather we have a distribution for our parameters of interest. And this is really rich because we can again use a point estimate such as the median of the distribution if you want to summarize our knowledge of that parameter, but we also have access to the uncertainty which comes in the shape of the distribution, right? And this is I really think this is powerful, really powerful, because we can use that uncertainty as input for other part of models in other questions, which is essentially what happens when we use calibrations in divergence time estimation. We are essentially translating what we know or the uncertainty on what we know into a new analysis, which is again telling us things in terms of uncertainty. And how happened to work in musicology is because the system was just perfect. I mean, we had Gregorian chant, which is some form of melodic music associated related to the mass, right? So we essentially have melodies which sort of behave like DNA sequences and are encoded with symbols, with letters. So it was both sequential and it was discretely encoded. So it was pretty simple to use the idea of aligning them, and aligning them makes sense. So they have parts which remain constant, but parts which are varying. So we use multiple sequence alignment and then Bayesian methods for topology estimation so that we could build a tree of the sources that record those melodies. For more details on that, I have some works already published or submitted, which are available as preprints and over my Google Scholar or my website. You can find the
Yeah, sure.
citations if you're interested.
Sure. Thank you so much, Gustavo. And I think we can also discuss in daytime. So I think
Thank you very much.
with this we close the question and answer session. And thank you all the participants for being here. Thank you so much. And thank you, Gustavo, for your wonderful talk. And please fill out the weekly feedback form and link will be provided in the chat box, which help to us learn more about the participants. And next week, we will be having another talk on November 25th at the same time and same day. And we will be hearing from Margot Nelson from Calvert Marine Museum, USA. And he will be presenting on vertebrate paleontology. And next, we'll be having a tea time with our today's speakers. So to discuss in a like more relaxed setting, if you are really interested to discuss in little detail with our today's speakers. So stay around for the tea time. And before that, Before that, we will be having a break for 2 minutes. So drink water and get your coffee or whatever drink you want and freshen up and come back, and then see you in 2 minutes.
Thank you very much, everyone.